Good morning, good afternoon, and welcome everyone. Thank you for joining us today for this new webinar session. I am Géraldine Sicot, business developer at CleanCells, and today we'll be discussing about biological products in a veterinary context, and the scientific expert with me is Marc Mechnin, CSO and one of the co-founder of CleanCells. So just a little housekeeping before we get started. So we'll be running 2 sessions of questions and answers during the webinar. So if you have any questions, please type them into the chat window on the right-hand side of your screen, and I'll bring them up during the presentation. And if you miss anything, don't worry, a recorded version of this webinar will be available online via a link that we will send to you within the next days. So let's start first with a short introduction of our company CleanCells, and Marc will then continue going through the main guidelines to driving the production and characterization of biologics in a veterinary context. So we know also that the European Pharmacopoeia has changed a year ago, so then we will discuss about the consequence for your products, and we will end this webinar with some case studies. So who we are. CleanCells is a French CDMO CRO, and we can offer you a high-level quality services for the development, production, and/or characterization of your biological products. We are specialized in process development and validation, GMP cell and virus banks manufacturing, as well as the characterization of these banks by our QC department. We also have some storage capacity, and we are able to develop or transfer some methods and to do the validation for further routine GMP testing. So those services can be applied for different types of products, such as antibodies, recombinant proteins, vaccines, and cell and gene therapy, as well as bacteriophages. So CleanCells was created 20 years ago now and was GMP certified in 2014. We are now more than 100 people who are handling more than 1,000 GMP studies per year, which correspond to more than 5,000 samples analysis. And we have about 20% of our employees dedicated to the research and development, with the main goal to develop and validate new analytical tools, and to customize some current methods. We have different profile of customers, which are located all around the world, mainly France, Europe, and US. And they can be biotech or big human pharma, as well as CMO or veterinary pharma. And we observed that the veterinary market is increasing since 2 to 3 years and seems to be promising in the future. A last word about the group. Clean Biologics, created 3 years ago. So CleanCells and more recently NowBios are both part of this group. So both companies are independent, but with complementary activities. For vaccine product development, we are both able to manufacture GMP master and working cell bank, and as well as virus bank, depending on your requirement for the grade of production. So NowBios can then take in charge the virus purification and virus inactivation process, as well as the process development and optimization of this process. And NowBios will then transfer this process to GMP and will proceed with the scale-up of the production to produce the final product until the lot release. While CleanCells will support you and every step, which is the QC expertise for raw material, in-process, or final products. And CleanCells can also transfer and validate some specific tests needed for the final release. So I will now let Marc going through his presentation focused on veterinary products.
Thank you. Hello, everyone. My name is Marc Mechnin, and as a scientific director at CleanCells, I am used to deal with regulatory requirements in the veterinary field. Saying that, I think it is good to start by questioning who makes the rules, who makes the guidelines. So here in this slide, I focus on the main ones. On one hand, the European part with the EMEA and the EDQM through its EP chapters. On another hand, the FDA chapters and the US Pharmacopoeia. VICH aims to gather and harmonize the regulatory requirements. But keep in mind that you may have to comply with other regulatory requirements, for example, those from Australia, Japan, and/or China. So it is worth to think about the market you want to address. So coming back to the European side, there are three main chapters in the EP which address the veterinary field. The first one is the 5.2.4, cell cultures for the production of veterinary vaccines. Another one is the 5.2.5, management of extraneous agents in immunological veterinary medicinal products. And the last one, the 0062 chapter, which is for the vaccines for veterinary use. You may also consider the EMEA guideline on requirements for the production and control of immunological veterinary medicinal products, and a specific reflection paper which address the different methods that are found suitable to perform the different tests. With regards to the US guidelines, we mainly have to deal with the 9 CFR guidelines, which are generic ones and quite old ones. We also have the common guidelines, OIE-WAIS, for the World Animal Health Information System, which is in fact the equivalent of the WHO for the human field. Now, let's go through the European chapters and the main changes that occurred in July 2020. In fact, a big change is the complete removal of chapters 2, 6, 24, and 25, which were initially dedicated to avian vaccines and used to describe in details the different approaches to test for avian viruses. For instance, use of primary cells, permissive cell lines, and so on. Besides this change, chapters 5.24 and 5 and chapter 0062 were deeply modified, even rewritten. Well, keep in mind that the goal to achieve remains the same. We want to make sure that cell substrate used for the production of vaccines are free of contaminants such as bacteria, fungi, or viruses and retroviruses. The main change in this chapter is that there is no recommended approach anymore. There is no mandatory strategy. This strategy should be defined by the sponsor on the basis of supportive data like internal data, literature, reflection paper, or any kind of data that could help. The risk analysis will help to determine which tests are needed and will allow to cover the lists of contaminants described in chapter 5.2.5. If we look at this slide, we can observe a specific table that applies on a cell substrate and what is expected at the different stage of the production process. So mainly master cell seed, working cell seed, and cells from working cell seed at highest passage level. We can see the main item or type of tests that are needed depending of the step in the lifespan of the starting material. We need to check for the karyotype, the identity, the absence of bacteria, fungi, or mycoplasma, the tumorigenicity, especially if no data is available, and finally, the detection of viruses and retroviruses, which is the aim of the chapter 5.2.5. Once again, if we look at the chapter 0062, the goal remains the same. Make sure that the virusid, which is the basis of the future vaccine, is free of contaminants. Like for the cell substrate, the strategy of testing was suppressed and should be defined by the sponsor on the basis of any supportive data. A risk analysis should be conducted so that we can cover the list of contaminants described in chapter 5.2.5. Now, let's talk about this chapter. I already mentioned a lot from the beginning, the EP chapter 5.2.5. First of all, it is worth to notice the changes that occurred in the title with the introduction of the word management, which is in line with the expected work. We have to manage the detection of contaminants to give the insurance of safety for the starting material. An emphasis is brought on the risk analysis, including the risk assessment, where the material comes from. For instance, is there any kind of virus or specific virus present in the country of production? The risk control, does the process contribute to contaminants elimination? And control measures, especially the detection tests. This chapter also introduces the use of molecular biology tools, and by now it's fully accepted. Besides this, a list of contaminants to be detected dependent of the species origin of the starting material and the targeted species for the intended use of the vaccine is introduced. Indeed, previously we used to look at the EMEA guidelines to determine the list of contaminants we had to test for. Nowadays, the list has changed. Some contaminants were removed, some emerging ones were added. The list is now part of the EP and covers all the following species. The avian ones, with generic test, and additional ones, respectively for chicken, duck, goose, pigeon, or turkey. There are also some viruses for bovine, porcine species, equine, canine, feline. In the case of rodent, you should address the mouse, the hamster, the rat species. There are also some guidelines regarding the primates, and especially the viral cells, which is largely used in the production of vaccines. And very recently, we had also the introduction of fish viruses with salmonids and finfish. So in this slide, we have decided to present only one example, which corresponds to rodent species, and more precisely, the rat species. You can see here a list of viruses, of bacteria that you need to test for. And you can also observe that endogenous retroviruses should be also tested.
Thank you, Mark. We will go ahead and take time for questions now. It seems that the European Pharmacopoeia is the most detailed guidelines in terms of list of tests to be detected, as you exemplified with the rodent one. So can we consider that if a product is European Pharmacopoeia compliant, this product will be also FDA and other guidelines compliant?
Okay. As we have seen, the requirements coming from the EP is quite huge. From my experience, and after discussion with a representative of the USDA, I would say that the product will be likely compliant with other guidelines. EP is classically considered as covering all needs. We just need, in fact, to check for any kind of specificity for a given territory.
Okay, and so we have a question about the MARA, the Chinese Pharmacopoeia. So is there any specificity that our attendees should know about it, except what you already said about?
Well, to be honest, I have recently had to deal with this authority, but it was also the case with Australia, in the context of a specific project. I would say most of the requirements, especially virus to be detected, are the same. However, the design of the test may differ from one guideline to another guideline. Thus, it is important to discuss and to convince the authorities that our tests are equivalent.
Okay, so we have more questions on the regulation. We will maybe keep going to keep our timeline, but we will go back at the end if we have time to all the questions on the regulation. If we do not have time to take all the questions, we will answer at the end of the webinar by regular email. So let's move with the case studies and general approach for adventitious agents and retroviruses detections.
So we went through the theory. I think it's useful to go into case study to make it more concrete. On this slide, we present both types of starting material, cellular samples and virus seeds. Our approach here is the following. In case of cell substrate, we apply first a 20-day culture period of the sample to allow the expression of potential contaminants. Then, as far as possible, we try to use a CPA detection approach using permissive cell lines. The advantage of this approach is, in fact, it's a non-specificity and the broad range of viruses that can be detected. Additionally, for viruses that can be amplified on permissive cells, but without any CPA demonstration, a specific detection, especially by PCR, can be performed. Finally, for those viruses for which no cell model is known, the approach will be to perform direct PCR on the material generated after the 20-day culture period. In case of virus seed, the first question we need to ask is, do we have an antiserum available? If the answer is yes, then the approach will be nearly the same as for cell substrates with the use of permissive cells. The only difference will be the culture period, which will be 20-day long instead of 14 days. If no antiserum is available, then we will have to proceed with a PCR approach. In this slide, we focus on the case of retrovirus detection. If you remind or remember the table and the list, we have always to address virus detection and retrovirus detection. So it's a more generic approach for the retrovirus detection. If we consider this detection in the context of cell substrates, there are two possibilities. First of all, if no RT activity is expected, no non-endogenous activity, then Q-paired should be performed. If an RT activity is expected, then we can go directly through a permissivity assay with the Q-paired assay after 21 days of co-culture. If now we want to test virus seeds for these retroviruses, we will likely face cell lysis, and consequently, we will have to deal with DNA polymerase in the samples, and consequently, RT-like activity. In such a case, a first approach will be the performance of a Q-paired assay without and with thymus DNA. This thymus DNA will quench the non-specific RT activity. Depending on the results, we could either conclude, if negative, or think about an infectivity assay. Now let's talk about a first case study. Here we are in the case of a rodent cell line used for the production of a porcine virus. We will have to deal with all items presented in the table on the right. First, a general microscopy is required, and will consist in morphological description, and you will have to take some photographs. Additionally, a karyotyping analysis on 50 metaphases will be performed. The key point here will be to determine the ploidy level and their abnormalities, the modal chromosome number, and the gross structural abnormalities. In complement to this karyotype, we will have also to perform a species identification. A common approach by now is the use of PCR for this topic. We need then to confirm the absence of microbial contaminants, and this will be done through a sterility assay, which is a very common test. This will be also the case for the mycoplasma, which is addressed either by PCR, direct or indirect culture. With regards to tumorigenicity, as previously discussed, this assay should be conducted in absence of previous knowledge on cell line status. After this consideration to generic items, let's go back to the detection of viruses and retroviruses. Here we have a CHO cell line or a BHK21 cell line, so coming from rodent, with the swine species for its final use. We will first cultivate the sample for 28 days. Then this material will be used to inoculate indicator cells, chosen for their ability to show CPE in presence of, for instance, porcine coronavirus or mouse encephalomyelitis virus. These cells could be also chosen for their ability to amplify a number of viruses without any CPE, but before performing a PCR test. For example, that could be applied for the detection of PCV or PPV. Several viruses will be tested by direct PCR because of the absence of suitable cell models for their amplification. This is the case for hepatitis E virus or African swine fever virus. On this next slide, we have chosen to present the approach applied for the retrovirus detection. Here, it is well known that these rodent cells express endogenous RT activity. It is then necessary to determine if this activity is due to infectious retroviral particles or not. In this aim, an infectivity assay on a specific cell model, mousse Denis cells, is the best approach, since this cell model is known for its ability to amplify murine retroviruses. A QPERT assay after 21 days of co-culture will allow to conclude. The second case study corresponds to a virus seed produced on porcine cells and for porcine targets. Once again, sterility and mycoplasma should be addressed. With regard to virus detection, a key point is the availability of antiserum. If yes, and after determining suitable conditions for the neutralization, we can get back to the use of permissive cell lines and the detection by CPE, for example, here the Nipah virus, or by PCR after amplification on cells, for example, once again, PCV or PPV. Alternatively, in absence of antiserum or in absence of suitable cell model for virus amplification, PCRs can be performed. Here, the slide is to emphasize on the difficulty we may face in the context of the testing for the presence of retrovirus, especially for porcine species. As mentioned before, if there is a cell lysis, non-specific RT activity is likely. And we may overcome this issue by using thymus DNA spike. Here we also have RT activity coming from the cell substrate itself. Thus, thymus DNA could be insufficient to eliminate RT activity signal. The expected approach is therefore to conduct an infectivity assay. However, at the time being, it appears that no porcine cell line has been described to be fully retrovirus-free, and therefore a suitable cell model is still under investigation.
Okay, thank you, Mark, for this second part of this webinar. So we will take again one more time for the questions now. So there is one question about the amplification step for the step of amplification for 20 days for the extraneous agent detection. So can you please explain the relevance of this step, if it's really necessary, or if they can be skipped, or because it's a long step?
Okay, that's true. In fact, I am convinced it is better to have a high quality material to perform the detection of contaminants. Therefore, an amplification step appears as a good approach. Moreover, if we look at the 9 CFR, it is required to apply at least a 21-day culture period. So 28-day culture, which used to be the standard in the previous EP guidelines, remains very convenient.
Okay. Again, around this step, these 28 days plus the 14 days after with the co-culture plus the PCR detections. And if a client on this product, on his product, has only a few viruses to be detected because he did his risk analysis, so what would your advice, for example? Can we go straight for the PCR detection on the product, or still better to go all these culture steps?
Okay, the quick question here is probably the amount of materials that will be submitted to the tests. In other words, this is a matter of representativeness and sensitivity of the test. As you can imagine, the two steps approach with an amplification of the potential contaminants on the cells, and then a detection by PCR is better. Nevertheless, you may consider, with regards to any kind of timelines, to apply the PCR, but it's really to be linked to the sensitivity of the assay.
Okay. Another questions is the clients are the specific target to be detected. Can we adapt the test to be able to support his need?
In fact, with the new list of viruses and corresponding species, we may have to deal with the absence of cell models and/or antiserum. That's why it is important to develop new tools for the testing, such a new PCR. I'm proud to say that CleanCells is here to assist its client for their specific needs.
Okay, thank you. I'm just looking for other more questions. Let's see. So there is one question going back to the regulation. So the question is there any ATMP guidelines as well in the vet medicine?
To be frank with you, I didn't have the opportunity to really work on that topic. What I do know is that we have some cell therapy, for instance, and classically we base our test on the chapter I already mentioned.
Okay, thank you, Mark. And we are looking for more questions. So the last one maybe, so can we justify the absence of tumor regenicity test if no cells are detected in the final product?
Well, it's really a good question that corresponds, in fact, to the risk of transfer of specific DNA sequence into the animal. So, from my side and from my opinion, this can be justified like this, but once again, it's really a discussion with your authority.
Okay, thank you, Mark. Maybe a last one about the risk assessment. So as far as I understand what you said during the webinar, the risk assessment is under the client responsibility. So on our side, for example, we are just then doing the detection of the virus or contaminants that were selected by the client, right?
Yeah.
Okay. Yeah. Thank you, Mark. All right. There are more again. So yes, there is another one. Sorry for the timing. So I can't see the full question on my screen. Sorry about that. So yeah, but this one is yeah, what guidelines to follow for innovative biologics. I mean, it's related to the ATMP that we already talked about. And this one, yeah, in case of cell bank consisting of suspension cells, do you recommend to go directly to detection of cytopathic agents? But we do not have the end of the question, so we will go back to the client by a regular email. Sorry for that. Yes, we will now maybe go to the key takeaways. Sorry, the main message that we wanted to talk with you today, Mark.
Sorry, first of all, for not answering all the questions, but we will do afterwards.
Sure.
We have a couple of issues with regards to the questions. Well, in fact, regarding the key takeaways, the EP chapters in the field of veterinary vaccines have deeply changes. So you have all the time to consider these new chapters. The risk analysis is a central point when building the strategy of testing. That means that you may avoid to test for some contaminants if there is a clear I ought to say that if it's clear enough that these contaminants cannot be a part of your vaccine, for instance. The species origin of the starting material and target species must be taken into account to establish the list of tests to be performed. And finally, and once again, it is very important to well define your testing strategy to make sure to cover all the needs.
Okay. Thank you very much, Mark. We saw, for sure, we will go back to all the questions that we missed, and we will then answer you by a regular email. So that's you.
I can just see one question, so maybe just very quickly. Yeah, for bovine polyomavirus, there is clearly no infectivity assay feasible because no cell model was described. So clearly the PCR remain the key approach.
So then if there is a positive sample, it's
it could be a bit tricky, but that's the way it is. But you know, till now we didn't face this issue. I would say we may face this kind of issue for instance for the BVDV, for which it's really necessary to perform a first step of amplification using indicator cells and then perform a PCR just to make sure that there is nothing coming from you know nucleic acids remaining in the sample itself.
Okay, thank you. And the last question is still a pop-up. Yes, we have a catalog for all those PCR available at Clean Cells, so we will communicate on this to the person that asked for. Well, that's all we've got, I guess, for you here today. So thank you again for joining this webinar. We appreciate you signing in and hope you learned a lot. Before you sign off, I'd love to tell you that we will run a new webinar session in December or January, so feel free to reach out to me via email if you're interested or want to know more. And I hope that we will see you next time. Bye-bye.
Thank you. Bye.